Alright, welcome everyone to our video on the plasmid isolation and purification experiment. As you should have read in the laboratory manual, plasmids are circular pieces of DNA that can be copied and read by bacteria, and we use them in the laboratory to perform molecular cloning. Again, as it says in your lab manual, molecular cloning is the process of cutting and pasting DNA together. We can use plasmids, which again are circular pieces of DNA, cut them in order to insert foreign pieces of DNA, which the bacteria can then copy and transcribe into proteins, which we can then easily study in the lab. What we're going to do today is to show you how we go about making, isolating, and purifying plasmids. Now there's two ways plasmids can be stored in the laboratory. One are at minus 14 degrees in liquid samples. But the most common and stable way to store plasmids are actually inside bacterial strains. So we keep bacteria strains that have copied these plasmids at minus 80 degrees C, and then whenever we need to make more of that plasmid, we simply allow the bacteria to grow, and as the bacteria grows, it makes more copies of that plasmid, which we can then isolate. Now what we're going to show you first is how we actually go about growing the bacteria that contain the plasmid. Now these plasmids have they have the code for antibiotic resistance genes. The reason for that is because the bacteria actually don't want to keep and replicate the plasmid because it takes energy for the bacteria to do so. In order to force the bacteria to keep making copies of that plasmid, we grow the bacteria in the presence of antibiotics, and those plasmids have the genes the bacteria need to help them resist the effects of those antibiotics. So, in order to survive in the environment, the bacteria has to keep making copies of the plasmid, which has those antibiotic resistance genes, and we can then ensure that all the bacteria we grow have the plasmids we need. Now, let's go ahead and get started. As we mentioned, we're going to be growing the bacteria in the presence of antibiotics. The first thing we're going to want to do is sterilize our area because we're going to be working with live bacteria. So I'm simply going to use 95% ethanol to sterilize the surface on which I'm working. And I'm going to be working under the makeshift sterile conditions where we have a flame here. So this is a dangerous protocol, but we're using proper laboratory precautions. This flame is going to create an updraft and help keep our environment sterile. So the first thing we're going to do is we're going to add the antibiotics to our growth medium. In this case, the antibiotic we're using is streptomycin. And so we're going to take our pipette, this one here, set it to the appropriate volume. We're simply going to take our antibiotic stock solution, and under our sterile environment, we're going to add that to our growth medium. We'll give that a stir to make sure our antibiotic is properly dispersed. And now we have our growth medium with streptomycin present. The next thing we need to do is to dispense this growth medium into the culture tubes that we're actually going to use to grow the bacteria. To do that, we use our sterile pipette. As you can see, it's singly packaged to make sure that it stays sterile. We withdraw the appropriate amount of growth solution, or growth medium, and we're going to pipette 5 milliliters into each of our 2 culture tubes. Perfect. Now we can set these aside. We have our culture tubes with our appropriate growth medium. The last thing we need to do is to add our bacteria. We have here our bacteria, which are stored in glycerol at minus 80 degrees C, and that basically allows the bacteria to remain cryogenically frozen, and they'll come back to life as soon as we add them to our growth medium. So we simply take our pipette, the appropriate volume, we're going to take 5 microliters of the bacteria, add it to our growth medium. Change tips each time, ensuring the conditions remain sterile. And there we have it. Now we have 2 culture tubes, 5 milliliters each, both of which now contain our bacteria. We're We're going to let these grow overnight where the bacteria will replicate and create very thick cultures. And then tomorrow, we'll be able to isolate the plasmid DNA that is contained within these bacteria. Alright, so now our bacterial cultures that we inoculated have grown overnight, and as you should be able to see, we now have two very confluent or thick cultures of bacteria. All the cells that are in these solutions contain our plasmid, and so now we simply need to break open the cells and in order to isolate and then purify the plasmid. So the first thing we're going to do is collect our cells. In order to do that, I'm simply going to pipette the cells using our large pipetter into an epi-tube. And then we're going to place the epi-tube in the centrifuge and spin it for 1 minute. Of course, with an appropriate counterbalance in place. This is set for 1 minute. We're keeping it cool to make sure the cells are just fine. And we're doing this at 13,000 RPM. So that'll spin for 1 minute. A little bit of an old centrifuge, so it likes to make some noises. As you can see now, we have a nice little pellet of cells that have been centrifuged, and they've clarified our growth medium. So the next step is to empty out our growth medium. We'll use our pipette to make sure that we get rid of all of the growth medium. And now it's on to the step of breaking open the cells to release the plasmid. The first step, now these procedures are performed with commercial kits. In this case, we're using the Monarch Plasmid kit provided by New England Biolabs. And they provide us with a series of solutions that are going to make our job very easy. Now the first step is to resuspend that pellet. And so we're going to take 200 microliters of the resuspension solution, which you can see is a nice pink color. We're going to pipette it into the epi-tube, and then we're going to repeatedly pipette up and down to resuspend that pellet so that it's not stuck down at the bottom and the cells are again floating around in the solution. Now we have a nice pink solution with our cells resuspended. We're finished with this solution, so we'll set it aside. And then we're going to move on to use our lysis solution. Lysis is our fancy biochemical word for breaking open the cells. Essentially, this solution is a detergent, and that will break up the cell membranes. And so we're going to pipette 200 microliters of this nice blue solution into our epi-tube. Cap that, and we'll give that a few inversions. And we can already see, it's hard to see here, but the solution has gone from being cloudy to rather translucent. That's because the cells are being broken apart. We'll wait about 30 seconds for the lysis to take place, and use that time to clean up our workstation. Now the next step is going to be to precipitate out the proteins and lipids. Of course, there are many more components to the cells that we're breaking apart than just the plasmids that we want to isolate. There's going to be lots of membrane lipids and lots of proteins in there as well. So in order to isolate the plasmid, we want to precipitate out or eliminate all of those proteins and lipids. Luckily for us, New England Biolabs provides us with a precipitation buffer. And so we're going to add 400 microliters of this solution to our lysed cell solution. Simply pipette our 400 microliters, add it to our lysed cells, and we'll give that a few inversions as well. Now what you should be able to notice is that this solution is no longer translucent. It's no longer translucent, it's full of chunks. Those chunks are again the precipitated proteins and lipids. Now we're going to give that about a minute to continue its precipitation process, and then we'll simply run the centrifuge once again in order to eliminate all those precipitated proteins and lipids. Good. That looks good to me. So I'm going to go ahead and load it again into the centrifuge with the appropriate counterbalance in place. And this time we're going to run the centrifuge for 2 minutes. Alright, so now our centrifuge has finished, and as we can see, all of those proteins and lipids have now been pelleted out of the solution. And we're left with a clear yellow liquid that contains the plasmid that we want to isolate. Now we're going to move over to our next step, which takes advantage of this very important piece of equipment that we call a spin column. As you can see, this spin column has a white membrane, and that membrane is designed to bind only to the DNA in our liquid. So what we're going to do is load our liquid into the spin column and centrifuge it so that the liquid passes down through the membrane. Any plasmid that is contained within our solution will stick to that membrane. So I'm simply going to take my pipetter, draw up the yellow liquid, of course with our plasmid. I'm then going to load it into our spin column. And we're going to walk over and once again centrifuge this such that the yellow liquid passes over the membrane. We'll set that for 1 minute. Alright, now as you can see, our yellow liquid has passed through the spin column, and all of the plasmid DNA that was in that yellow liquid should now be loaded onto the membrane. We'll come back to our workstation. We'll discard the liquid that has now lost its plasmid. Now, as we said, when we pelleted the sample, we should have gotten rid of most of the proteins and lipids and things we don't want. But we need to make sure that our sample is completely clean before we wash the plasmid off of the membrane. So we've got 2 wash steps that will allow us to get rid of any other junk that might be stuck on the membrane along with the plasmid DNA. So our first wash step calls for 200 microliters of the wash buffer to be added onto the spin column. And once again, we're going to centrifuge that for 1 minute so that this washing buffer passes over the membrane to get rid of any junk that might be present. I'm going to switch our balance, and we'll again run this for 1 minute. Alright, now our first wash step is complete. Again, you can see the washing buffer has passed over the membrane. We discard the washing buffer, And we're going to add our second washing buffer. This time we're adding 400 microliters as prescribed by the kit's instructions. Once again, we will load that into the centrifuge and spin it for 1 minute. Alright, now our second wash step is complete. Now what we have is a very pure, clean sample of the plasmid on the membrane. We've gotten rid of all of the junk from our 2 wash steps. So now what we're going to do is take a fresh epi tube and we're going to stick our spin column into the epi tube, as you can see here. We're going to use a final special buffer that is called the elution buffer in order to wash the plasmid off of the membrane. As we said, this membrane is designed to stick to the plasmid, but we're going to use a special buffer that is designed to release that plasmid from the membrane. We'll use 35 microliters of this special elution buffer. We will load that onto the membrane very carefully, and we will wait 2 minutes for the buffer to solubilize the DNA. By waiting 2 minutes, it helps the buffer sort of wash that DNA off of the membrane. Alright, so now we've waited our 2 minutes. The buffer has taken effect and it's ready to wash the plasmid off of the membrane. We're going to load that into the centrifuge. And we're going to run the centrifuge, again with an appropriate counterbalance, for a total of 2 minutes. Alright, now our centrifuge has completed. You can see from looking at the sample that we have a small 35 microliters of the elution buffer that has traveled through the membrane, and now our concentrated plasmid DNA is all contained in this 35 microliters of elution buffer. We started with a cell culture, and we finished with a nice, clean, pure sample of plasmid DNA that we can go ahead and use for molecular cloning or whatever other applications we have. Alright, welcome to the second part of our experiment. Now, as we talked about from our previous experiment, we often use plasmids, our circular DNA that we isolated, in molecular cloning, where we insert foreign DNA into those circular DNA vectors. Now, when we do molecular cloning, it can sometimes be challenging to figure out if the experiment went correctly, because of course DNA is such a small molecule that it's very difficult to see. So how do we go about finding out if our molecular cloning experiments actually work? Well, that's what we're going to be doing this week. We're going to be doing what's called a restriction digest. Now, restriction digest uses enzymes that have evolved to cut DNA at very specific sequences. What we're going to do today is take our plasmid that we isolated, which has not been used for molecular cloning, and we're going to take the result of a molecular cloning experiment, and we're going to do a restriction digest using 2 restriction enzymes. Those enzymes will cut the circular DNA on either side of where our DNA has been inserted through molecular cloning. If the molecular cloning experiment was successful, we will see that inserted piece of DNA when we analyze our samples using DNA gel electrophoresis. So we'll do a restriction digest on our control, our isolated plasmid, which we will not see any inserted DNA. And we will also do the digest on our sample which we've used for molecular cloning, and if we see that insert, we'll know that the molecular cloning was successful. Our first step in that process is going to be to set up our restriction digest reactions. In order to do that, we're going to mix water, our plasmids, the restriction enzymes, and a special buffer that is designed for these types of experiments. So the first component of our reaction is simple. It's just sterile distilled water. I'm going to take the water and add it to both our control experiment and to our unknown molecular cloning experiment. Next, we're going to add the special buffer, which is actually called Cut Smart buffer. Of course, it's called Cut Smart because it's used for restriction digest experiments. We're going to add 5 microliters of the Cut Smart buffer to both our control and our unknown experiments. Changing tips each time to ensure our buffer stays nice and clean. Great. Next, we're going to add the plasmids that we want to cut in this restriction digest experiment. We will add 10 microliters of those. First, our control plasmid. And next, our unknown plasmid. You'll notice we're setting up these experiments using these very small, thin-walled tubes. They're very small because, of course, these reactions are a total volume of only 50 microliters. And they also have a very thin wall, and that allows heat to easily transfer from the incubator, where the reaction will take place, into the solution itself. Alright, so we've got the buffer, the water, and our plasmids that we want to digest. The final thing we need to add are our restriction enzymes. We choose those restriction enzymes carefully so that they cut the circular DNA exactly where we want them to. In this case, we're using 2 restriction enzymes, the first being XHO1, and the second being BAMH1. We add 1 microliter of each enzyme to each experiment. First, I will add the XHO1 to our control experiment. Changing tips again to make sure our enzyme solutions stay nice and clean. And now I will add the XHO1 to our unknown experiment. Cap that, and we can move on to our second restriction enzyme, this being BAMH1. Add the BAMH1 to our control experiment. And then I will add the BAMH1 to our unknown experiment. Now we've got everything we need in both of the reactions. And the final step is to simply mix gently these reactions to make sure the enzymes are thoroughly distributed throughout the solution. I'm going to set my pipette to 10 microliters. I'm going to simply draw the solution up and down slowly and gently, perhaps 10 or so times, ensuring that the solution is thoroughly mixed. That looks good. I will, of course, change my tip. And I will do the same for our unknown experiment. Now these restriction enzymes, the ones that are cutting our DNA, have been engineered to operate at 37 degrees Celsius. So our next step is going to be to take these reactions and put them in an incubator at 37 degrees Celsius. Now, these enzymes are engineered to operate very quickly, so these digests can actually take place at that temperature in just about 15 minutes. We're going to ensure that those digests occur completely and that there's no undigested plasmid left. So So we're going to leave them for about 1 hour. So we will take our reactions, we'll put them in the 37-degree incubator, and we'll come back in 1 hour, and then we'll be ready to analyze our samples. Alright, so here we have our 37-degree incubator. That is a standing incubator, meaning it's not going to shake. We'll simply take our rack that has our reaction tubes in it, we'll place it in the 37-degree incubator, close the door, and then we let it rest for 1 hour. Alright, so now we've had our reactions incubating for 1 hour. We'll take the samples out. Alright, so in order to analyze our restriction digest, we're going to perform a technique called agarose gel electrophoresis. We all know that DNA has a phosphate backbone with a negative charge. And so we can apply a charge to our DNA samples, and they will actually move away from the negative charge and towards a positive charge. We're going to get them to move through a gel because that gel has very small pores. The smaller the DNA fragment, the easier it will move through the gel because it will be easier for that small DNA fragment to move through the small pores. That's the idea of agarose gel electrophoresis. The smaller the DNA, the faster it will move through the gel. The larger the DNA, the slower it will move through the gel. This is the technique that allows us to separate DNA fragments, and that's going to allow us to analyze the products of our restriction digest reaction. Now, in order to perform agarose gel electrophoresis, we have to actually make an agarose gel. To do that, we need to take our agarose, which is actually a purified sugar polymer, and we will mix it with a special buffer called TAE. We will heat that up. That will melt the agarose, and then we can pour that melted gel into a mold. So the first thing we need to do is to measure out the appropriate amount of agarose. For the gel we are making, we need 1 gram of agarose. That'll do nicely, close enough. Clean up our mess. Now, we're going to take that agarose and pour it into our designated flask that we use for making agarose gels. That's 1 gram of agarose. And because this is a 1% gel, we're going to add 100 times the volume in TAE buffer. 1 gram, 100 mils of the buffer. That's going to give us a 1% agarose gel. Give that a little swish to make sure the agarose is fully, not soluble, but suspended properly. And there you can see we have our buffer and our agarose, but clearly it is not melted. So we're going to take it over to our very high-tech laboratory equipment that we call a microwave. We're going to put it into the microwave, plug the microwave in. We're going to heat it for about 1 minute. As it's heating, of course, the sugar polymers will start to melt and solubilize in solution. Alright, so now as you can see, our solution has become translucent, or transparent, excuse me, and all of the agarose has melted. Now that solution is still very hot. We are going to be adding a dye called ethydium bromide to this solution, which will allow us to actually visualize the DNA that is run through the gel. However, if we add the ethydium bromide while the solution is still very hot, we would expect that dye compound to perhaps decompose. So what we're going to do is give this about 15 to 20 minutes so it can cool down to till it's warm. Then we will add the ethydium bromide and we'll be ready to pour the gel. Alright, so now our agarose gel solution has cooled until the point where it's warm to the touch, and so we're ready to pour the gel into the gel mold. Now first thing we're going to do is construct the gel mold. Here we have the gel plate. You can see it has rubber on the outside, which will allow it to seal to the mold so that it does not leak. So we're going to simply push this into the mold so it creates a tight seal. And we push it all the way down. And there you can see there's a chamber now where we're going to pour our gel. Now, as I mentioned before, we have to add ethydium bromide, which is our stain, to the gel so that we can actually visualize the DNA in the gel. This is a very important point because ethydium bromide is highly carcinogenic. It's a very toxic compound and it's known to cause cancer. Why does it cause cancer? Well, it works to help us visualize the DNA because it sticks. It's a flat molecule that can insert itself in between the base pairs of DNA. If you get it on your skin or in your mouth, it can do the same thing to your DNA, which can cause mutations, which of course lead to cancer. So we need to be very, very cautious when we're working around ethydium bromide. That simply means being very aware of our surroundings and of course wearing the proper protective equipment. We're going to take our ethydium bromide solution using our pipetter. We will draw up some of that solution. We will add 1 drop to our agarose gel solution. Return the rest to the bulk sample. And then we simply need to swish our gel around so that the ethydium bromide is distributed throughout the solution. I'm going to cap the ethydium bromide. Now we have our gel. It has the dye in it. And we're going to pour that into the mold. We're going to pour it slowly so that we don't get bubbles all over on the inside of the gel. And finally, we're going to add the comb. The comb is going to create spaces in the gel where we can actually load our DNA samples. Of course, we need somewhere to put our samples into, and this comb is going to create the spaces for those DNA samples. You can see on the mold there are spots for the comb to be inserted. We insert the comb there, and there we have it. The gel is ready to cool. As it cools, it will harden, and after about 15 minutes, it will be hard enough for us to actually load our samples and then to run the gel. Alright, so as you can see, the gel that we poured is now successfully hardened. Our restriction digest reactions are complete, and so we're ready to load these DNA samples into our electrophoresis gel. We made the mold in the same chamber that we're going to actually run the electrophoresis in. So the first thing we want to do is remove the comb, which as we said, has created the spaces where we can actually load the DNA. I'm removing the comb by very slowly and gently pulling straight upwards, and that will ensure that none of the spaces between the wells are disrupted. As you can see, there are now spaces for us to load the DNA. I'm going to turn this way. I'm going to remove the mold with the gel in it and simply turn it 90 degrees. And now it is placed in the chamber where the electrophoresis can occur. The next step is to take the same buffer we used to make the gel, that being the TAE buffer, and to fill up the chamber with that buffer. We fill it up so that it's just covering the top of the gel. Perfect. Now we have a chamber with a gel that is perfectly ready for electrophoresis. The next thing we need to do is prepare our samples. Now, as you might imagine, if we were to take our restriction digest samples and directly load them into these wells, they would float away because this sample is no more dense than the buffer that is currently filling the wells. In order to ensure that the samples don't just float away and that they happily sink to the bottom of the wells, we're going to add what's called a loading dye. It's a nice purple color and it's very dense, so when we mix our sample with the loading dye, it will sink to the bottom of the wells. We're going to add 20 microliters of the loading dye to each of our restriction digest samples. I will add it first to our control reaction. And I will pipette up and down so that this dense loading dye is evenly distributed throughout the sample. As you can see, our samples are now a nice purple color. And I will do the same to our unknown sample. All right, now both of our samples are ready to be loaded into the gel. Now the wells that we made in the gel when we poured the gel can hold between 40 and 50 microliters of solution. So what I'm going to do is add 40 microliters of each of these samples to the wells in the gel. To do that, I'm going to make sure both of my elbows are very stable because this requires a little bit of technique. I'm going to load the 40 microliters into my pipette. I'm going to ensure that the pipette tip is submerged in the well, and then I'll very slowly eject the solution into the well. As you can see, that purple sample happily settles to the bottom of the well. I'm going to leave a space that we'll talk about in just a moment, and on the other side of the space, I'm going to load the control sample. Again, we see the sample weighed down by the loading dye settles happily into the well. Now, we can run these two samples and we'll be able to compare them very nicely. However, we want to add one additional component, which is called the DNA ladder. The DNA ladder, which I have here, consists of a variety of sections of DNA that are all of known size. By running our samples next to the ladder, which has many pieces of DNA which have a known size, we can compare the DNA sizes in our samples to the DNA samples that are in the ladder. That will allow us to get a definitive size for all of our DNA samples. I'm going to take 10 microliters of the ladder, and I'm going to load that into the well between our two samples. The ladder already has loading dye in it, so it naturally settles just as our samples do. Now we have all three of our necessary wells filled up. We are ready to actually run the gel. Now, in order to do that, we need to set our power source to the appropriate values. We're going to set it to a voltage of 100, a current of 400 milliamps, and we're going to run this for 60 minutes. So I'm just going to push the up button until we get to 60 minutes. Now, to run these gels appropriately, we're going to keep the voltage constant. So you can see the power source has the voltage set at a constant level. We're then going to take the top of our chamber and attach it, ensuring that the red side is away from our samples. Remember that red means positive and black means negative. Because DNA has a negative charge, it's going to want to move through the gel away from the black or negative electrode and towards the red or positive electrode. If we attach these opposite, the DNA will run backwards and off of the gel, which of course we do not want to happen. Now our chamber is all hooked up. Our settings are set appropriately on the power source. We simply press the run button. That will start the current flowing, and we should be able to see bubbles begin to course from the electrodes. If we can see bubbles, that means the current is running and our DNA will begin to travel through the gel. Alright, so as we can see, our gel has now completed its 60-minute cycle. I'm going to take off the top of the chamber, and you can quite clearly see that the loading dye has traveled about half to 60% of the way through the gel. Of course, the loading dye is not DNA, but that tells us that the DNA has also moved through the gel as we apply the current to it. The last thing we have to do then is actually analyze or have a look at our gel. So I'm going to very carefully take the mold, of course using gloves, out of the chamber here. I'm going to just let it drip off so I'm not getting buffer everywhere. I'm then going to transfer the gel from the mold onto our UV light table. I mentioned that ethydium bromide is a dye that allows us to visualize DNA. Well, this dye fluoresces under UV light. So in order to actually see the DNA, we have to use UV light, and that's why we have this special UV table. In order to see it, though, we're going to have to turn out the lights. Now, very strong UV light, as you all know, because you know you shouldn't stare at an eclipse, can damage your eyes. This UV table, then, has a protective cover that will actually protect your eyes from the damage of the UV light. So when we visualize these types of gels under UV light, we only look at them through these protective coverings. I will then turn the table on, and there we have it. We have our DNA samples. We can see from our unknown sample, there are 2 bands, and from our control sample, there is only 1 band. This is what we expected, because we know that the control sample does not have any DNA inserted into it through molecular cloning. However, our unknown, we did not know if there was any DNA inserted through molecular cloning, but what we can see from this electrophoresis experiment is that there is indeed an additional piece of DNA that has been inserted into our plasmid. This tells us that our molecular cloning experiment has been successful. Now, what is this extra piece of DNA? Well, in most cases, it's foreign DNA coding for a protein. Molecular cloning, as you have read about, is a technique that biochemists use to study proteins. From this experiment, we have a circular piece of DNA that now contains a foreign gene that codes for a protein. We now know that E. coli can actually produce this foreign protein because of our molecular cloning experiment. This protein could be anything. It could be an enzyme that we're interested in using to perform reactions. It could be a protein that is involved in cancer that we're interested in studying in further detail. But it's a protein that could be from any source, which we can now easily produce using E. coli that we can easily grow in the lab in large quantities. Through this molecular cloning experiment, we created a piece of DNA that E. coli can read and produce a protein from any source which we can then study. This is the magic of molecular cloning.